The goal of this lecture is to understand how coupled carbon and nitrogen models at ecosystem and global scales may be represented in matrix form. Basically, nitrogen transfers among all organic nitrogen pools can be represented in one matrix equation that is equivalent to the carbon matrix equation. The carbon and nitrogen matrix equation are coupled through the carbon and nitrogen ratio. Mineral nitrogen dynamics determined by the nitrogen input, mineralization, plant uptake, and the leaching can be also described by one equation. Rising atmospheric CO2 concentration tends to include carbon sequestration in terrestrial ecosystems. The conceptual framework of a progressive nitrogen limitation has predicted nitrogen limitation on future carbon sequestration in terrestrial ecosystems in response to rising atmospheric CO2. The nitrogen limitation may become progressive stronger over time unless nitrogen fixation is stimulated or nitrogen losses are reduced, leading to increased nitrogen capital. In addition, the degree of nitrogen regulation on terrestrial carbon sequestration depends on changing several carbon-nitrogen coupling parameters, such as the stoichiometric flexibility of a C/N ratio of biomass compartments. Changes in plant nitrogen uptake via soil exploration and the nitrogen redistribution from the soil to vegetation. Encoding what we know about how carbon and nitrogen flow within ecosystems, how these flows are coupled, and can help to fully understand the strengths of the nitrogen regulation on carbon sequestration in terrestrial ecosystems. One of the coupled carbon and nitrogen models presented in this lecture is developed from TICO model. TICO has 8 carbon and nitrogen pools in addition to 1 mineral nitrogen pool. Carbon enters the ecosystem through canopy photosynthesis and is then allocated into foliage, wood, and fine roots. Similarly, nitrogen is absorbed by plants from mineral soil and then partitioned among leaf, wood, and fine roots. Detritus from plant biomass turnover is transferred to metabolic litter and structural litter pool. Where it is decomposed by microbes, the structural litter is partly respired, while partly converted into fast and slow soil organic matter. During these carbon cycling processes, nitrogen in plant detritus is also transferred among the same set of ecosystem pools. Mathematically, these carbon processes can be described by the first-order ordinary differential equation. See the blue equation, where X represents the carbon pools in leaf, wood, fine root, metabolic litter, structural litter, microbe, slow, and the passive soil organic matter respectively. C is the vector of environment scalars accounting for the temperature and the moisture effect on all carbon decomposition. A describes carbon transfer formation among various ecosystem compartments. Matrix A describes carbon transfer coefficients. K is the baseline turnover rate of carbon pools. I is the input and B is the location of inputs into ecosystem carbon pools. Similar to carbon, the nitrogen processes can be described by the pink equation. The dynamic of the mineral nitrogen pool is determined by balance between nitrogen input and output through the plant uptake and the nitrogen loss, which can be expressed by the third equation, where the first term is the rates of nitrogen uptake and loss. The second term on the right side of equation 3 describes the amount of nitrogen release during mineralization. Phi is the proportion for mineral nitrogen production. The carbon and nitrogen cycles are coupled through the parameter R, which is a C/N ratio. F represents nitrogen input through biological fixation and atmospheric deposition. To illustrate the application of matrix forms of a CN-coupled model, we designed a case study to examine changes in CN coupling parameters under CO2 enrichment using a data simulation approach. Based on measurements of carbon and nitrogen pools in various ecosystem compartments and the fluxes obtained from Duke phase experiment, key parameters of TiCo-CN were estimated through a Bayesian probabilistic inversion. The inversion was done separately for plots with ambient versus elevated CO2 treatments, yielding one set of parameters for each. Comparison of parameter distributions showed that the plant nitrogen uptake, C2N ratio in foliage, vine root, metabolic litter, and structural litter was significantly higher under elevated than ambient CO2, whereas CO2 enrichment didn't exert significant effect on C2N ratios in wood tissue and soil organic matter. Moreover, elevated CO2 led to the decrease of carbon exit rate in foliage, wood, structural litter, and passive soil organic matter, indicating the increase of carbon residence time in this ecosystem component. By contrast, elevated CO2 resulted in the increase of carbon exit rate in vine root, demonstrating faster vine root turnover under CO2 enrichment. In addition, carbon allocation to foliage becomes smaller under elevated CO2, while carbon allocation to vine root tended to be larger under CO2 enrichment. The estimated parameters were then used for forward analysis to examine ecosystem carbon and nitrogen dynamics under ambient and elevated CO2 conditions at Duke Forest. Our results demonstrated that modeled nitrogen pool in foliage, wood, vine root, and the forest floor closely match with the corresponding measurements for both ambient and elevated CO2 scenarios. However, TiCo-CN couldn't capture the observed declining trend of the microbial nitrogen content with time. In addition, the trained model didn't simulate nitrogen dynamics in mineral soil well, partly due to the large variation in soil organic matter measurements among different years. Parameter of a C2N ratio in foliage, wood, vine root, metabolic litter, fast and slow soil organic matter, plant nitrogen uptake, and external nitrogen input were well constrained within their prescribed range under both ambient and elevated CO2. However, the nitrogen allocation coefficient to foliage, the rate of nitrogen loss, and the initial value of mineral nitrogen pool were poorly constrained. In addition, C2N ratios in structural litter and the passive soil organic matter and the nitrogen allocation coefficient to wood hit edged the lower limit under both ambient and elevated CO2 scenarios. This bar chart represents maximal likelihood estimators of carbon and nitrogen coupling parameter under ambient and elevated CO2. Arrow bars represent standard deviations of parameters. Different letters show significant differences. Comparison of parameter distribution showed that plant nitrogen uptake, C2N ratio in foliage, vine root, metabolic, and the structural litter was significantly higher under elevated CO2 than ambient CO2, whereas CO2 enrichment didn't exert significant effect on C2N ratio in wood tissue and soil organic matter. Relative difference in modeled total ecosystem carbon pool in the Duke Forest using two sets of parameters. First, those CN coupling parameters derived from observations under ambient CO2 are used to predict carbon dynamics under CO2 enrichment. For comparison, CN coupling parameters estimated from observation under elevated CO2 are also used to predict carbon dynamics under CO2 enrichment. Then the related difference in model total ecosystem carbon pool is calculated to illustrate the effect of the changing CN coupling parameter on model prediction. The insert is a box and a whisker plot showing the relative difference in model total ecosystem carbon pool using two sets of parameters. Total ecosystem carbon pools in the Duke Forest under CO2 enrichment was underestimated by 7.8%. If TICO-CM model parameters at ambient conditions were used to predict carbon dynamics at treatment scenarios, it further showed the cumulative nature of the relative difference in total carbon pool, indicating that underestimation would become much greater, especially for typical long-term predictions. Now we are going to extend the ecosystem scale to global scale. We implemented the matrix representation into a global land surface model, the Community Land Model, version 5. The vegetation biogeochemical module simulates the biogeochemical transfer among 18 carbon pools and 19 nitrogen pools. Tissue, storage, and transfer pools are included respectively in leaf fine root, leaf stem, dead stem, leaf coarse root, and dead coarse root. In addition to carbon pool, nitrogen pools include one more retranslocation pool temporarily storing nitrogen from later fall. All these biogeochemical processes and pools in vegetation modules can be formulated as carbon or nitrogen balance matrix equations. The soil biogeochemistry module has 20 soil layers. Each layer contains 7 pools for organic carbon and organic nitrogen, respectively in metabolic litter, cellulose litter, lignin litter, coarse woody debris, fast soil organic matter, slow soil organic matter, and passive soil organic matter. Inorganic nitrogen pools such as ammonium and nitrate interact among each other or with organic carbon and organic nitrogen pools, or with the environment through multiple nitrogen processes such as nitrification, denitrification, leaching, atmospheric nitrogen deposition, and biological nitrogen fixation. Non-zero entries in these matrices represent the carbon or nitrogen transfer among different pools. For example, the vertical mixing coefficient matrix V is usually a tridiagonal matrix, which means carbon or nitrogen in the soil only transfer between adjacent soil layers. All these biogeochemical processes and pools in soil module can be formulated as a carbon or nitrogen balance matrix equation. Carbon and nitrogen cycles in original CLM5 model are fully preserved in the matrix version. To check whether matrix equation works correctly, the carbon and nitrogen storage from the matrix model was compared with the original model. Carbon and nitrogen storage in total ecosystem, vegetation, and soil organic matter from the matrix model matched well with those from the original CLM5. It is worth mentioning that the matrix model is not an exact representation of the original model because different processes in the original model are updated stepwise, while all processes in matrix model are updated simultaneously. However, our validation results show that the relative differences of soil carbon and nitrogen are less than 1% for most grid cells. Only 0.4% and 0.5% of the grid cells in vegetation carbon and nitrogen storage respectively diverged by more than 1%. See the figure on the right-hand side. Global annual means in all 6 carbon and nitrogen storage over 115 years perfectly line up at the 1-to-1 line, signifying exact agreement. See the figures on the left-hand side. The good agreement demonstrates that the matrix model can faithfully reproduce both temporal dynamics and the spatial patterns of carbon and nitrogen states from the original models. In addition, matrix representation of a biogeochemical module provides an important diagnostics to understand model results. For example, the matrix representation enables the traceability analysis. Traceability analysis can be used to understand the carbon dynamic differences which result from different models, meteorological forcing, parameter schemes, and scenarios. We will learn this in following days. Generally speaking, we attribute differences in carbon storage into carbon input, environment scalars, and baseline residence time. Let's see a simple example. COM5 had a problem on global steady-state soil carbon storage. The global soil carbon storage modeled under 2 meteorological forcing can be different by over 100%. The global soil carbon storage are 2,500 gigaton carbon in GSWP3 versus 5,000 gigaton carbon in CRU-NCEP. Results from CRU-NCEP simulation had a significantly higher soil carbon storage in permafrost region. The highest carbon storage can be over 250,000 gram carbon per meter square, which is substantially larger than the GSWP3 simulation. Without further information, it's hard to identify the source of the differences, since meteorological forcing variables only differ at most 10% between GSWP3 and the CRU-NCEP. The difference in soil carbon storage was primarily attributed to a much longer soil carbon residence time in CRU-NCEP simulations, which are 74 years, compared to 38 years in GSWP3 simulation. It's nearly double. However, the global soil carbon inputs were different by only 11%. Now we can focus on residence time. The carbon residence time can be further decomposed into environment scalars and the baseline residence time. Specifically, differences in total soil carbon storage can be quantitatively attributed to differences in carbon input, baseline residence time, temperature scalar, water scalar, oxygen scalar, nitrogen scalar, and other scalar respectively. Among all the traceable components, the water scalar consistently made the most significant contribution to the difference in soil carbon storage under the two sets of forcings. Differences in environment scalars have been further compared globally. The most significant differences are still found in water scalars, especially in permafrost region and desert. Both permafrost region and the desert have a little liquid water in the soil. In desert, soil carbon storage in GSWP3 simulation doesn't differ so much with CRU-NCEP simulation because carbon storage are zero, with zero carbon input in desert. In permafrost region, tundra provides soil with a persistent carbon input, so carbon storage can't be zero in permafrost region, but carbon residence time will be very sensitive to the water scalar. As a result, carbon storage can be significantly different in permafrost region.